we would like to welcome Sarah Teichmann to the question and answer session. Thank you very much for the seminar, Sarah. It's very comprehensive with some great visuals. And to the audience, questions can be submitted via the portal. And along with Mathew and Rosa, our co-chairs, we'll answer as many of these questions as possible. So one of the first questions that has come in, Sarah, is that for each tissue atlas, are tissue samples from more than one person used? And are you able to account for factors such as age, sex, lifestyle, on cell type and composition?
Yeah, that's a great question. And the answer is yes. For many tissues, we're now at the point where aggregating the information across the community, so not from any one particular lab, but taking samples together from, let's say, the lung biological network of the human cellulose community, we have samples from hundreds of different donors. And so that does allow us to deconvolute covariates such as gender. And in fact, this is what we did recently as part of our work on analyzing ACE2 expression in cells from the airways. And what we found was increased ACE2 expression with age, for instance, gender, and change in ACE2 expression in the bronchi versus the parenchyma, dependent on smoking status. And you can read about that on our bioarchive manuscript. It's first author is Moose, M-U-U-S et al. And it's a consortium authorship from the HCA Lung Biological Network on covariates in relevant for COVID-19 from our human cellulose lung data. So that's not true of every single tissue at the moment. Some are more deeply studied and charted than others, but we are starting to get to the point in the community where we can put together data and identify covariates.
Thank you.
Hi, Sarah. I'll follow up with the second question. I might just continue on a bit from that, because you mentioned looking at different expression of things like ACE2. And I was wondering if that gave you any insights into the different, in terms of the effect we see with different ages, and particularly within children who seem to have much less severe consequences of COVID-19 infection.
Right. So that's those are exactly the questions that we addressed. And so with age, there's an increase in expression of the receptor in the parenchyma and bronchi, suggesting that the cells are more easily infected. And in fetal and neonatal and pediatric tissues, there's much lower expression, suggesting that there's less chance for the virus to dock onto those cells, the epithelial cells.
Is that universal across different tissues, or is that specific?
No, so the lung is a particularly well-studied tissue. There's been a lot of interest in the airways from labs around the world. And that's why we had enough data. And because of the COVID-19 pandemic, there was also a very rapid pooling and sharing of data in the community in terms of the metadata, the donor metadata, and the expression matrix prior to publication. So in a way, that tissue is probably a year or two ahead of other tissues. And I should say that very open and very rapid data sharing in the human cellulose community was precipitated by the COVID-19. So we don't have nearly the same number of samples. I think that's probably fair to say, either published or unpublished, for, let's say, the liver or the kidney or something like that.
Yeah, thank you.
Yeah.
Thank you. So following in the COVID project, I was wondering if you have been able to do nuclei RNA-seq data or to generate RNA-seq data from infected lung alveolar cells to really interrogate the cell death pathways that are active?
So the samples from significant epithelial tissue from infected patients is really only available in large numbers from post-mortem samples, so from deceased patients. And there are efforts in several places in the world, in the UK. So at Sanger, we're collaborating with Imperial College, with Michaela Naseda, Brian Hanley, Michael Osborne, on studying post-mortem tissue. There are also efforts in the US in several locations, in Boston, San Francisco, and so on, where there are studies of post-mortem tissue. And Omar Baraktar here in Sanger is using spatial methods, including highly multiplexing a molecule FISH and nanostring GeoMX platform with whole transcriptome amplification to study the post-mortem tissues. And there are some really exciting results from those studies that I alluded to in the talk. And there are also efforts on studying these tissues from nuclear RNA sequencing from frozen samples that I'm aware of in Boston. And there are some early indications, indicating things like increased immune cell infiltration in the patient lung versus control lung versus healthy controls. There are, I would say on the cell death pathways, the spatial distribution is particularly interesting because the viral load correlates with a lower abundance of 82 cells. So they're very, very clear kind of microenvironmental correlates that you can see from the spatial architecture of the tissue.
Thank you.
We've got a couple of questions relating to the microenvironment and how the complex environment in a tissue may affect gene expression. Two of the questions are asking if you're able to unpack this within the human cell atlas.
So able to unpack, can you repeat it again?
Yes, it's just that the gene expression may occur vary due to the interaction of a cell with its microenvironment, and whether the human cell atlas work is able to start to understand the effect of the microenvironment on gene expression at the single cell level.
Yeah. So the spatial data that I mentioned, that the spatial transcriptomic analyses of human tissues are really extremely revealing in the sense that telling us about microenvironmental factors that determine cell fate. And I mentioned very, very briefly, very superficially, our work on the endometrium, and Roser is really the expert on this who's led that project. And one of the major conclusions from that work is that there's cell-cell communication that determines epithelial cell fate that differs between the basal layer of the endometrium, the glands, and the luminal layer. And so it's those stromal cells that are signaling to the epithelial cells that determine whether they become secretory cells in the glands or whether they become ciliated cells in the lumen of the endometrium. And it's that that combination of single cell resolution, defining the spatial location in the tissue and the microenvironment in the tissue, and the whole genome transcriptomic description of the cells that unravels those cell signaling events that control cell fate.
Thank you.
Hi, Sarah. So one of our most highly voted for questions is the following. What are the current and projected computational challenges associated with the Human Cell Atlas project? So both current and projected.
Mm-hmm, mm-hmm. So the computational challenges are really driven or correlated to a large extent with the developments in technology. And there are two major directions, or two or three. One being multi-omics, so multimodal measurements on the same single cell. For instance, epigenetics in RNA, DNA in RNA. and it's, you know, the integrated analysis of those multimodal data that you know, presents a challenge or an opportunity for development of new computational methods. The second major area are the spatial methods that are just developing extremely rapidly, which I alluded to in the talk. And I mentioned two different kind of approaches or directions there. One being the sequencing, and the other being the imaging-based ones. And both of those, you know, have their own computational challenges in terms of interpreting the data, ranging from the low level cleaning up of the, you know, processing of the data, QCing, normalizing, and so on and so forth. And then there's the integration of that spatial data with the suspension cell data or nuclear data or multi-omics data and so on. So I think there's it's a very exciting time for bioinformatics in that sense. Then there's other challenges that relate less to technologies and more to the biological questions. And those are things like connecting in vivo and in vitro data, you know, so, developing methods that address biological questions that come out of the sheer amount of data on different types of samples. Another one is, you know, what we just talked about earlier on the questions on covariates. There's just so much data now that there's scope for you know, calculating influence of covariates and that kind of thing. So yeah, so I would say there are some computational challenges that go hand in hand with the technology development, and there are some computational challenges that come out of the sheer amount of data on different types of samples. So new questions can be asked and new methods are called for to answer those questions.
Thank you.
So following in the multi-omics integration, there is a question here that asks, do you feel that there may be differential expression variation from cells that may be impacted by the somatic mutations or epigenetic changes due to cellular microenvironments? And if so, are they detectable in the data?
Great question. I point to our recent Cardellino paper in Nature Methods, where we relate somatic mutations from deep exome sequencing of the HIPSCI Human-Induced Pluripotent Stem Cell Initiative fibroblasts to their single-cell transcriptomic phenotype. So that's McCarthy et al. In Nature Methods recently. What allowed us to do that correlation between the somatic mutations and identifying mutations that correlated with increased proliferation rates of cells, of fibroblasts, was the full-length RNA sequencing data that we carried out, and then having the deep exome sequencing in parallel. And so we were able to call genotypes within a single line, so within the same donor, so intra-donor clonal variation. I think that's much trickier to do using standard tag sequencing, where you only have the 3' or 5' ends of the transcriptome, and you're covering less of the exonic, or coding regions of genes. Then it becomes much harder to call those somatic mutations. So it's just a pure technical limitation, but I think it's a great question. And having deeper data, full-length data eventually for suspension cells, on a larger scale from ex vivo samples, coupled with the deep exome sequencing, maybe even with spatial data, will be really fascinating to dig into that in more detail.
Thank you.
Yeah, for tumors, it's obviously a much simpler problem, because they are filled with mutations and changing very rapidly in their transcriptome. But I'm talking about kind of non-healthy tissue, basically, non-tumor tissue.
Hi, Sarah. There's a question here that's been voted up the list, and it's about the application of the data from the Human Cell Atlas and whether the knowledge could eventually lead to the growth of organs in the lab for applications such as transplants, and how far off are we from that type of application?
So whether data from the human cell atlas could be used to engineer or?
And yes, to engineer organs in the lab
I think that's
and use them for transplants.
Question. Yeah, I think that's a super exciting question, and one that we're really excited about. And certainly, as we, in fact, as Roser and collaborators from the pathology department, Margareta Turco, showed, is that the data can certainly be used to engineer more specific cell lineages and more faithful models in vitro to the healthy human tissue. Now, I think using those engineered tissues for transplantation is one step further. But certainly, there are cell-based therapies that are starting to be approved for clinical use, and we have collaborated on projects where in vitro engineered cells are repairing cells in the mouse and even in human surrogates and so on. So I think that the community is inching closer and closer to that regenerative medicine application.
That's brilliant. Thank you.
Hi, Sarah. So one of our more popular questions that's being highly voted is, how do you define a cell type or cell state? Are these discrete states or a continuum of different states?
So the human cell atlas data is showing us more and more relationships between cell states, and in particular transitions that can take place in different contexts. So it's a slightly, for instance, in wounding and wound healing, cells kind of take on transitions that they wouldn't usually take in a normal homeostatic context in a tissue. So the question is it's a slightly semantic question. I mean, cell type has traditionally been used for sort of the major cell lineages that have particular functional attributes and maybe particular developmental sort of ontological origins. But I think more and more we're seeing that in vivo there are lots of wild and wonderful transitions that take place. And of course, in vitro, you can engineer huge numbers of transitions between cells artificially. So I think it's also complicated by the fact that the word cell type is used in different ways in different fields. So in immunology, cell types are very tightly linked to their cytokine secretion profiles, for instance, because that determines key functions of immune cells. In neuroscience, the neuronal cell types are very much defined by their connectivity to each other, so the networking of the cells. So I prefer to kind of shy away from that and let the data tell us, what the relationships between cells are and what the major, the most abundant cells are and the frequencies and so on. And then basically avoid the cell type, cell state debate altogether, kind of from a statistical point of view. Yeah.
Thank you.
So the next question is related to the next steps of the HCA. So going forward, is the priority to achieve a complete human body organ coverage, considering the practical difficulties of get those tissues, for example, healthy brain, or is it to increase the number of donors from more easily accessible tissues?
That's a very valid and interesting question. I mean, I would like to point out that the brain is actually one of the first tissues, or one of the premier tissues, if you will, that has been atlased, at least to a first version, to a first draft, where the Allen Brain Institute has taken a lead on the adult brain and very nicely linked the single cell nuclear sequencing data to the MRI defined and the different regions of the brain, at least for certain regions. I mean, not a complete map yet. So the brain isn't actually sort of very behind in that way. As I mentioned with the lung, there are, yes, there are accessibility kind of determinants that have contributed to some tissues being more studied than others, and also kind of biology and disease determinants. And so the lung is one, you know, where there's a huge amount of interest. From my point of view, so we've divided, we've sort of, the Human Cell has been a very grassroots-led and scientist-led bottom-up initiative, and we've over the past sort of half year or so, we've divided the community, or the community has kind of divided itself into biological networks. And if you go to humancellist.org/coordinators, you'll see the 12 or so major systems where there's kind of convergence of communities. And of course, Roser leads the reproduction biological network with others. And I'm very involved in the Immune Cell Atlas, the immunity biological network, and studying lymphoid tissues and immune cells. And you know, there's a very exciting group focusing on human development. There's another group looking at organoids, another group dedicated to human genetics, and that really relates to diversity of samples, which is most accessible for blood. So there's a kind of very broad human genetics, human variation initiative in the domain of blood, you know, because of accessibility. So I would say the Human Cellist community is going both deep and broad, kind of simultaneously in different areas.
Yeah, thank you.
Hi, Sarah. We're just reading the questions as they arrive. There's a question asking about, beyond transcriptomic readouts, what are the other state-of-the-art sequencing-based technologies, and can they be multiplexed in the same cell?
Yes.
And what additional value do these add?
So that's what I was referring to earlier with the computational challenges on the multi-omics. And I think I mentioned DNA, you know, epigenetics and open chromatin, RNA. Increasingly, there's also plexity in terms of antibody, oligonucleotide conjugated antibodies that can be sequenced at the protein level. So there's you know, very exciting multimodal technologies looking at many different levels, methylation of DNA as well. You know, we'll be looking forward to RNA modifications at some point, yeah. So there's much more than RNA. One of the ones I'm particularly excited about is open chromatin, because of the link to transcriptional regulation, transcription factor binding, enhancers, and so on. It's also very exciting from the genetics point of view, because as we know, a lot of the GWAS associations are non-coding regions that can be fine mapped to enhancers, essentially, to transcription factor binding sites. So that will, you know, be extremely interesting in the point of view of understanding that human genetic variation and those associations in more detail.
Hi, Sarah. We are nearing the end of the time, but if we could sneak in one last question before we close. I guess reflecting on the 5 years of the HCA now, what have you seen as the advantages of doing this work under a big umbrella of the HCA versus, for example, a much more kind of individual lab-led initiative?
So the HCA is a kind of interesting hybrid, you know, between work led by individual labs and consortium work, because it's essentially a sort of a community of many volunteers that are coordinating with each other. And the benefit that I see from that model, which is really a very new model, I think, for genomics consortia, and one that Aviv and I, you know, when we started 4.5 years ago or so, we felt very strongly that we want to have that model, is that it leverages people's individual interests and expertises. It leads to, there may be some replication, which leads to consolidation. And then eventually, in the case of the lung and the COVID-19, as I mentioned, it then opens up new doors in terms of the covariates that we discussed, age, gender, and so on, smoking status, and so forth, in terms of cell types and their gene expression. So what I think the one weakness perhaps is the completion. If you have individual people who are working together all on their own research area of interest, how do we have sort of blind spots, and can we plug them? And I think as long as we as a community work together on that and keep track of that roadmap of getting towards a complete atlas, then we will reach completion and plug all the gaps sooner rather than later. Certainly this project is kind of, it's not even 4 years old basically at the moment, and it's there. If you go to the data coordination platform, there's millions of cells from over a dozen different organ systems. So the rate at which it's progressing is really pretty spectacular, and I think that sort of validates that community model for a genomics project. Yeah.
Thank you.
So we would like to thank Sarah once again for her presentation and for joining us for the Q&A session, and also the audience who joined this virtual session. So there will be a seminar each month, so please look out for announcements. So next month, the speaker will be Peter Campbell, who is the head of the Institute of CASM program. So his seminar will be about somatic mutations in normal tissues, and how is this providing insights into tissue development and maintenance, cancer evolution, and other diseases and aging. So thank you for joining us, and we look forward to seeing you next month.